Good afternoon, everyone. My name is Kilian Kleemann, and I would like to share today my research and my passion to tackle Alzheimer, and hopefully to contribute to our efforts to those who are affected. So to give you a short overview, as you can see here, this is an illustration of the vertical cross-section of a human brain. On the left side, you can see the anatomy of a healthy brain, which contains millions of these brain cells called neurons. On the right side, you can see the typical anatomy of an Alzheimer brain, which is shrinking. So the smaller size of the Alzheimer brain is due to the progressive loss of neurons, which is also described as neurodegeneration. Alzheimer's becomes more prevalent after the age of roughly 65. And this diagram here illustrates the percentage of people out of the whole world population who are 65 years and older. And previous estimates show a steady increase from roughly 5% in 1950 to now almost 10% in 2020, and further projections show a rapid increase. In fact, the incidence of Alzheimer's is expected to increase at a doubling rate from 50 million to almost 100 million in 20 years by 2040. Yet, we have no way to treat this disease, and recent drug trials have actually failed. So in order to approach treatments for Alzheimer, we need to understand how the disease starts and then how it progresses. Based on our current understanding right now, it starts with the abnormal accumulation of proteins into so-called aggregates, or kind of clusters or clumps of proteins. As we age, then, more of these proteins start to accumulate in and surrounding the neurons, which ultimately becomes too much for the neurons, and some of them will start to die. This is when neurodegeneration starts. However, it is not until much later that the first symptoms start to show, such as memory loss, and this is already when cognitive decline starts to worsen. So think of this as a type of house on fire. So the protein aggregation is the spark that allows the fire to grow unnoticed inside of the house. However, as soon as we see the flames coming out of the side of the house, it is already too late when the fire has spread too much. So in summer 2018, I was able to investigate certain molecules which help us to detect Alzheimer early. And interestingly, we not only found that these typical protein aggregates show that a person might develop Alzheimer, but also other molecules which are important in our natural injury response. In fact, now, this injury response to so-called inflammation has actually been shown to be a crucial part of Alzheimer progression. So what is inflammation, and why is it such a part of Alzheimer? On this slide here, I just want to give you a simplified illustration of the inflammation process and the role of immune cells within that. So here you can see two neurons which normally communicate with each other. But as soon as these proteins start to aggregate, they interfere with the neuron's function and they become stressed. So they send out stress signals, which are then recognized by a specific which are then recognized I had pressed the wrong button. Which are then recognized by certain helper cells or immune cells called microglia, which then actually start to chew up these protein aggregates. As more proteins start to aggregate, these immune cells then send out signals to recruit more of these helper cells or microglia. This process then is called inflammation. So a sustained stress signal during Alzheimer's can actually lead to some of these immune cells to become different. They become so-called hyperactivated, which essentially means that they not only start to chew up or eat these protein aggregates, but also start attacking the neurons themselves, which ultimately can lead to more neuronal death and more activation of the inflammation. So essentially, what I want to illustrate here is this sort of self-increasing positive feedback loop, which over time then leads to a sort of imbalance between the immune cells helping and the immune cells becoming overreactive and causing more damage. So I wanted to find out exactly how do these immune cells or microglia respond or know how to respond to these stress signals. And if we can improve our understanding of the communication between neurons and microglia, is there a way for us to re-establish this balance? So here you can see, again, the neuron and the microglia. Last summer, I was able to do research at Strathclyde University, where I investigated a very particular part of this communication between the two cells. So again, proteins start to aggregate, causing stress to the neuron, which sends out stress signals. On the microglia are specific receptors which recognize the particular stress signals. And once the signals bind to the receptor, it sort of activates a relay or cascade inside this microglia. So the microglia then knows how to react and what to do. In various different cells, it's been shown that some of these receptors can form dimers, which is when two receptors stick together. And these dimers have been implicated with a sort of refined or different response. What I wanted to find out whether these dimers are also present in microglia. And essentially what I did was to grow these microglia and expose them to different levels of the stress signal. Then I mushed up the cells and was left kind of with a solution which contained millions of different proteins. So for each stress level concentration then, I separated the proteins according to size, with larger ones on the top and smaller ones on the bottom. And to only visualize the specific receptor that I was looking for, this one here, I used a detection molecule or light-emitting detection molecule. So the light here is represented by these whiter bands. And at a particular size, we can see that with increasing stress signal concentration, there is more of this receptor present in microglia. However, we can also see at a higher size, so above that, potentially there is more of these dimers present. So these results were only very initial, and further tests were then carried out at Strathclyde University to find out more about it. And I started to think about how can we combine this idea of trying to regain the balance of the injury response and this idea of a precise communication between neurons and microglia to approach new treatments for Alzheimer. So essentially, the idea is to develop a more dynamic and multi-step treatment. So more precise, so an early detection and a more precise understanding of the communication will then help us to work together with the immune cells to eliminate or extinguish this early spark, thereby preventing the protein aggregation from reaching this critical threshold of neurodegeneration. Following on from that is a sort of continuous surveillance of the inflammation level, which hopefully, with the aim essentially to balance the helping microglia compared to the hyperactive microglia, and thereby preventing this hyperactive loop from spiraling out of control and causing more neuronal death. So essentially, with other long-term strategies, such as nutrition, cognitive exercises, but also sleep, we might then be able to reduce the cognitive decline. And I'm certain that increasing efforts today to find better detection and surveillance markers, as well as a more precise understanding of the communication, will help us and facilitate our efforts to work more dynamically with the nature's toolkit. So most certainly, my thinking has been inspired by our generation's fate of inheriting a house on fire, and this hope and ambition to restore a sort of balance and work in conjunction with nature, and in this case, natural processes. Restoring the balance of the injury response and moving towards a more dynamic and multi-step treatment will hopefully then help millions to keep sharing their memories and improve their quality of life. However, this will require a change in our understanding as well as our attitudes, away from a one-fits-all superdrug to a more dynamic and personalized treatment. Dynamic treatment. Sorry, I'm just having a blank spot right now. Oh yeah, sorry. So this approach is very unconventional. However, I think it is on our and the future generations to approach these growing challenges with ambitious, forward-looking, and creative thinking. Overall, I would like to thank the supervisors in the labs as well as the Biochemical Society for funding me to do this research, as well as the brilliant minds presenting before me and the organizers of this inspiring event. If you want to keep up with my research, follow me on Twitter at Kilian Kleemann, and I hope I started to stimulate your ambitions to become all world changers. Thank you very much.